Cancer is the emperor of all maladies, from which breast cancer is the second most dangerous and widespread cause of death in mankind. In 2021, around 27,700 Canadian women were diagnosed with breast cancer, making it about 25% of all new cancer cases. This statistical data shows why it is very important to find a sustainable solution to cure cancer. The main aim of the study is to analyze the effect of capsaicin on cell viability, cell behavior, apoptosis in MDA MB-231 cells and CHOK1 cells, and also to check the beta-catenin gene expression as it is responsible for the cell-to-cell adhesion in cancerous cells. Capsaicin is a chemical compound found in chili pepper, which belongs to Capsicum genus of plants. Many studies showed there are many beneficial properties of capsaicin, such as anti-cancerous activity, anti-inflammatory properties, antioxidant properties, etc. The investigation started with checking the cell viability at different doses of capsaicin-treated cells via MDT assay. The results showed the decrement in the cell viability with an increment of capsaicin dose in both the cell line, as shown in figure 1. Secondly, the cell behavior of cancerous cell and non-cancerous cells were analyzed via migration assay. The cells were treated with different doses of capsaicin and the results showed less migration of cells at highest capsaicin doses, suggesting decreasing its invasive potential of cancerous cell. Thirdly, acridine orange ethylium bromide assay helped in differentiating between live cell, apoptotic cell, and necrotic cells. The MDA MB-231 cells were treated with 50 micromolar and 200 micromolar of capsaicin for 4 hours. Then the dye mix were incorporated and the cells were viewed under different fluorescent filters such as GFP and Texas Red. Additionally, DAPI stains were also applied to validate the living cells from apoptotic cells. The results showed that the higher capsaicin treated cell led to higher number of apoptotic cells. Lastly, RT-qPCR was performed to analyze whether the beta-catenin gene gets upregulated or downregulated in capsaicin treated cells. Before proceeding to actual PCR run, the primer efficiency and annealing temperature was obtained as a validation. However, the experimental run didn't show significant downregulation of beta-catenin gene when normalized to GAPDH. There were some unexpected CT value appeared when optimizing the GAPDH primer, which led to discrepancy in gene expression results. Moreover, higher number of replicates can be beneficial to get a significant proof in downregulation of beta-catenin gene expression. However, one of the hypotheses was accepted as the cell viability, cell behavior decreases in both cell line in dose-dependent manner, and the apoptosis increases in the breast cancer cells with increase in capsaicin treatment. At last, capsaicin showed a remarkable results when treated with cancerous cells. However, with further investigation, capsaicin might be a vital element in the war against cancer.